I am Ms. Frida Pereira, Assistant Professor in Botany from Kamal College of Arts, Science and Commerce for Women, Nueva Goa. The title of this unit is Cell and its Components. Module name, Cell Membrane 2. Module number is 13. The outline of this module, the structure of the cell membrane. In this, we'll be studying about the fluid mosaic model. Next, the fluidity of the cell membrane. Learning outcomes. The student will be able to describe the fluid mosaic model of the cell membrane. The student will be able to explain the fluidity of the cell membrane. Okay, now coming to the structure of the cell membrane. The plasma membrane is formed of glycoproteins and phospholipids. Now, these molecules are arranged in definite pattern. The fluid mosaic model was proposed by S.J. Singer and G.L. Nicholson in the year 1972. According to this model, the plasma membrane consists of lipids and proteins. The lipid is in the form of fluid and the proteins are embedded here and there in the lipid in a mosaic pattern. Hence the name fluid mosaic model. The lipids are arranged in the form of two layers, outer layer and inner layer. The proteins do not form any layer. Each lipid molecule has hydrophobic tail and hydrophilic head. The hydrophilic head face outwards and the hydrophobic tails of two layers face each other. Okay, this is the image of the fluid mosaic model. Now the protein molecules are globular. Okay, and they are of two types, the peripheral or extrinsic proteins and integral or intrinsic proteins. The peripheral proteins are arranged on the surface and are loosely bound to the lipid. The integral proteins are deeply embedded and are tightly bound to the lipid molecules. The peripheral proteins as well as the outer integral proteins are stirred with sugars. Such proteins with attached sugars are called as glycoproteins. If extrinsic proteins were moved from the membrane, there is no disruption in the membrane. But if intrinsic proteins are removed, the membrane structure is disrupted. The sugars are also attached to the outer surface of some lipids. These lipids are attached sugars are called glycolipids. The carbohydrates found in intrinsic protein and glycolipids form sugary covering called glycocalyx. It recognizes certain foreign protein as immune cells and protects the cell from the extracellular digestive fluids. The fluid mosaic model stresses that the plasma membrane is semi-fluid in nature. The lipids as well as the intrinsic proteins move freely in the lipid bilayer. Next we come to the fluidity of the cell membrane. Now this fluidity of the cell membrane is based on three factors. First is the temperature, followed by the phospholipid composition, and third is the cholesterol. Now the fluidity of the membrane the optimal value of which is prerequisite for normal cell growth and function depends on the lipid composition and temperature. An increase in the temperature tends to elevate membrane fluidity. Lowering of temperature causes freely flowing, low viscous fluid membrane structure to be more rigid, gel-like organization. This change is described as phase transition, and the temperature at which this happens is called the transition temperature. Fluidity of the lipid bilayer also depends on the chain length and the degree of saturation of its component fatty acid residues. Lipid with short, unsaturated fatty acyl chains undergo the phase transition at lower temperatures than the lipids with long or saturated chains. Short chains have less surface area with which to form the van der Waals interactions with each other. Hence, a shorter chain length reduces the tendency of hydrocarbon tails to interact with one another. Unsaturated fatty acyl chains have nicks or cuts and tend to adopt a more random approach, which states from one or less stable van der Waals contact with the other lipids. So, increased proposition of unsaturated or saturated fatty acids in the membrane increases the fluidity of the bilayer at a reduced temperature. The next important factor is the cholesterol, which is embedded into the lipid bilayer. The cholesterol is also a major determinant of membrane fluidity. At high temperature, cholesterol interferes with the movement of the phospholipid fatty acid chains, making the membrane less fluid. But at low temperature, it interferes with interactions between fatty acid chains and prevents membranes from freezing. At the high concentration found in eukaryotic plasma membrane, cholesterol tends to make the membrane overall less fluid at growth temperature near 37 degrees centigrades. Okay, students, now to summarize the module, we have seen the structure of the cell membrane, that is the fluid mosaic model, and also which are the factors responsible for the fluidity of the cell membrane. These are the references for this module. Thank you.